Proteins, once you have a protein, a bunch of them together in a chain, there's the protein in a chain. We call the order of that amino acid primary structure. That is known as the primary structure. It's all written here if you're confused, but let me be very clear one more time. The order of the amino acids is called the primary structure. What determines the order of the amino acid? DNA. DNA, a gene, goes to mRNA, and we're going to go through that whole process, and then that will get you a specific order. So there's words here. These words mean something. These words make something, get translated, transcribed, and then translated into this sequence of amino acids. That's your primary structure. Why does that matter? Because if you have this order, maybe this protein, if let's compare it to hair, is straight because there's not a lot of cysteines. But if you have a bunch of cysteines in here, now all of a sudden you have curly hair. So it actually changes your phenotype. You remember phenotype and genotype from middle school, right? This actually is what changes your phenotype. That's how your genotype changes your phenotype. Because you change the amino acid order, you change the protein. You change the protein, you change how it works. So your secondary structure is when the side chains start to interact with one another. How do they interact? How can these side chains, how can these R groups interact? What can they do to each other? If you didn't have any way of knowing, I wouldn't ask you. What could happen between these things? What kind? Hydrogen bonds. Hydrogen bonds can start to occur. And depending on the order of amino acids, you get two different shapes. We call this a secondary structure. The secondary structure is determined by this primary structure. So you get, do you agree that this is two different shapes? One's helical or curvy or whatever, and the other one has kind of a stepladder kind of look to it. We call this a beta-pleated sheet. Beta is the Greek letter. And this is an alpha helix. Alpha helix. Alpha is a Greek letter. So this is kind of a shape, alpha helix. This is another shape. It's called beta-pleated sheets. So this structure is known as secondary structure. The shape is a secondary structure. What determines the secondary structure? Hydrogen bonds. Hydrogen bonds that are determined, whether you get hydrogen bonding or not is determined by the primary structure. So the primary structure determines the secondary structure. And so it flows. DNA or a gene gives you an mRNA that codes over here for a primary structure. That primary structure gives you a secondary structure, either one shape or another. It seems like it's a fancy word, but it's not. Secondary structure just means the shape. See there, it could be one of many shapes, but the two that you're learning about right now are what? Alpha helix, beta-pleated sheet. You can highlight it in your study guide and write it down in your notes. I think it's easy enough to draw it out, a little curvy line and a little stepladder line. So you can have a long protein that bends on itself. You know that. If you have curly hair, you know that because your hair is doing what? It's a long protein that does what? Curls up on itself. So one protein can hydrogen bond with itself. Tertiary structure. Every protein has a precise structure formed by folding a secondary structure. So you take this, you can fold it. Fold it and twist it and turn it, and you get a specific shape. So just going back to the one example that we all know very well because most of us have it on our head, hair, right? Everybody's hair has a specific structure, right? A shape, a texture. Some people are thick and some people's are thin. Some people's are long and straight. Others Other people's are kind of wavy, right? So it's not just curly or not, right? It's everything in between. So we call that tertiary structure. Here you get a lot of disulfide bridging happening. That's where the cysteines come into play. That's when you really start to get real tight curls. Someone close the door, please. We call that tertiary structures. Now, I've been talking about hair so far. I've been talking about hair because it's really the most common example. But let me be clear. Enzymes in your blood, hemoglobin also bends like this. And in fact, the difference between someone with sickle cell and someone that doesn't is one amino acid difference in this chain causes this hemoglobin not to bend correctly. And what ends up happening is you end up with a sickled cell, which causes sickle cell anemia. So that one amino acid change can change everything. Yes?
Should I go get some tissue?
Yes. So that's your tertiary structure. So you see, everything's built on everything else, right? Yes?
You said primary structure is determined by hydrogen.
No. Primary structure is determined by the peptide bond. The order of the amino acids is determined essentially by the gene. Initially by the gene. Which amino acid comes when? Now that's going to mean that you have different set of R groups, and each R group is going to have a different property, either acidic or basic or polar or nonpolar. And that's going to mean that either you're going to have a lot of hydrogen bonding or not a lot of hydrogen bonding. And that's going to determine the shape. So the secondary structure is determined by the primary structure. The secondary structure is determined by the primary structure. The primary structure is determined by the gene. Secondary structure is what force bends and twists these things into this shape? Hydrogen bonds. Between the R groups, which again is why it's important which amino acid goes when, because those R groups are going to interact differently depending on what they are. There's up to 20 different ones. Tertiary structure is a folding of the secondary structure. So you get these alpha, you see the alpha helix in here? You could also have a beta-pleated sheet in here, whatever. That will also bend because of disulfide bridging or some other reason. That folding, this is a very specific shape. I want to emphasize this. These shapes are important. Structure and function with proteins are directly related. So let me give you an example. If I draw this, and I draw this, which one would you use to enter a house? The door. Why not use this one? But why? And
Because it makes more sense. It's easier to use the door than it is to go through the window.
why is it easier? It has a what? It has a doorknob that lets you open it. It has hinges to let you swing it open. But if I had this, if this was the house, and you had little windows like this,
Oh, you might be fat. You can't fit through that.
I wouldn't fit through it.
I wouldn't either.
And all you had was a brick wall. Would you call this brick wall a door?
No.
Why not? So it doesn't work for entering a house, right? And either does a tiny little window. So this one works, the door fits the best, doesn't it? But why? Because of its shape. Its shape determines the structure. Why don't we make doors that look like, like this? Because you'd have to put the hinges over here, or over here, or over here, and either the door is swinging upwards and away from you, or it's swinging down to the ground. Either your doorknob is here and you're opening it downwards, or your doorknob is here and you're opening it up. That doesn't make sense to anyone. It's hard to do. Makes life harder. The functioning of a door doesn't work very well, does it? So over time, we've made the door rectangle. We could make it a circle, right? If you've watched Lord of the Rings, you know that they have circle ones. And it could open. You could put hinges here, and it could open. But it doesn't really work for entering and exiting a door, because when you do that, when normal human beings do that, maybe hobbits are different, but what happens is that people get tripped up on the edges, right? Circles don't work very well. You're trying to get something square through a circle, it can be hard to do. Something tall isn't going to necessarily fit through this circle unless it's a really big circle. So what we found through many, many generations of door making, we like rectangles. It works best. Now that's most places in the world. You go, I think it's fair to say most places in the world, you go there, you're going to find a rectangular door. The point here is not about rectangles or circles. The point is that the shape determines the function, right? The shape determines the function. This is true for molecules. Molecules also, their shape is determined by their shape determines the function. Your hemoglobin, if it has this shape, works great. That's the red blood cells. This is how you get oxygen to your cells. If this is broken, you're anemic. It's hard. You don't have a lot of energy. This is one form of anemia. Not everybody that's anemic has this. The sickle cell anemia is because this shape is off, so it can't carry oxygen well. And now you hear your red blood cells that are sickled. You're tired. Why are people with sickle cell disease tired so much? Because they don't have enough oxygen. They can breathe all day long, and they can't carry the oxygen because the shape of a protein in their cells, one single amino acid is different. The shape was different. So let's go down the list. They had a gene that made an mRNA that made a polypeptide chain that was slightly different. That small change made the secondary structure shape different, which meant that the protein did not fold correctly. And since it didn't fold correctly, it can't pick up the oxygen. Done. Anemia. Now, hemoglobin molecules are more than one polypeptide. So when you say a hemoglobin, this is the small difference between protein and polypeptide. A polypeptide is a long chain of amino acids, period. But a protein like hemoglobin is actually made of 4 chains. Do you see them? Here's all 4 of them. So in every one of your red blood cells, this is what it looks like. There's 1 iron in the middle of each chain. Everybody in here unless you have sickle cell disease, everyone in here has 4 chains, 2 alphas and 2 betas. 146 amino acids long for the beta, 141 amino acid long for the alphas. 2 different genes coding for 2 different polypeptides. There's actually more than one form of this problem. There's sickle cell anemia, that's a problem with one of the chains, and there's other diseases, there are at least one other, maybe a couple more. I don't know about the others, though. I would say definitely 2 that have, one is for this chain, the other disease impacts this chain. Again, if the shape is wrong, if this doesn't fold well, this thing cannot carry oxygen, which means you're tired all the time, and it's hard for you to survive. So alpha and beta chains. 2 identical alpha chains, 2 identical beta chains. This is called the quaternary structure. What is a quaternary structure? It's a structure or a shape that's made by the interaction of more than one polypeptide. So this shape, you see, is dependent on this shape, right? If this shape is different, then this thing is different. I mean, one thing leads to, it's like the dominoes, right? Like that, the domino effect. You hit one domino in a chain, what happens? The rest fall. So you change one little thing all the way up here in the DNA, and all of a sudden everything else changes down the line. All right. So we can change the shape. We can temporarily impact the shape of a protein. By the way, this is hemoglobin's chemical formula. There's 3,032 carbons in hemoglobin. That's in one hemoglobin molecule in your red blood cell. 3,032 carbons, 4,816 hydrogens, 872 oxygens, 780 nitrogens, 8 sulfurs, and 4 iron atoms. 1, 2, 3, 4. Now, when we say hemoglobin, we mean all 4 chains. So there's 4 polypeptides here. 2 genes, 4 polypeptides, leading to this crazy looking thing formula down here. And it only takes one amino acid, one small 3-carbon amino acid with this little side chain to be different for this whole thing not to work. That's how important every one of these steps is. Yeah.
So, the amino acids can only be different by a mutation?
Good question. Excellent question. Yeah, so what you're saying is amino acid sequence, it can be changed by a mutation. That is correct.
That's the only one?
Pretty much. There are mistakes that can happen, but mistakes that are not in the DNA, mistakes that happen during translation, they're going to happen at one protein or another. They won't happen system-wide, right? So, yeah, mutation. That's absolutely good connection. You see, he did what the medical student said you need to do. Make connections. So, here we have denaturation of proteins. So, it turns out that if you eat really strong, if you have, bless you, very strong alkalides, very strong acids, now not strong enough to break the covalent bonds, right? But strong enough to break, yes, go. Strong enough to break the hydrogen bonds, what would happen to the polypeptides? Well, if you broke the hydrogen bonds, not the covalent bonds. Oh, okay. What would happen to these things? They would separate. They would unfold. They would unfold. We call that unfolding denaturation. Write that down because that's going to be on the next quiz and it will certainly be on the next test. Denaturation. The unfolding, the breaking of the hydrogen bonds. The breaking of the hydrogen bonds, the unfolding of the polypeptides. You're disrupting the secondary, primary, tertiary, and quaternary structure. The primary structure stays the same because they're all covalently bonded. Those are peptide bonds. They're strongly held together. It takes a lot more to break these apart. You have to actually boil them to destroy those. But if you increase the heat slightly, if you increase heavy metals like mercury, if you put it, if you're exposed to radiation, UV light, heat, or detergents, or solvents, organic solvents, you can separate the hydrogen bonds. So this explains why you get sick, why you die if you get a bad fever. If you have 103-degree fever, why? You know what the doctors are going to do to you, what they used to do to you right away? As soon as you enter the emergency room, you come in with 104-degree fever, you know what they're going to do? Put you in a bathtub full of ice. They got to bring your temperature down. If they don't bring your temperature down, these proteins are going to unfold. If they unfold, all your what? What do proteins do? Everything. They do everything in your body. Everything's, your whole system stops working. So high fevers are not good over long, I mean extremely high fevers, obviously 101, 102 for a short period. That's your body's natural response. But a very high fever or an extended period of time with a fever can cause real damage to your body. Yeah.
So like, if like sun's small, and like, say like sun's small, right?
Hold on, I can't hear it because you're moving around. Go.
Say like sun's small, like not there, like that's gonna start to unfold, so it's not gonna look like that anymore.
That's right. If you will, any one of these four things happen, salts, heavy metals, strong acids or bases, heat, etc., can cause denaturation. Denaturation is the unfolding of the secondary, tertiary, or quaternary. Here's the cool part. The cool part is you can reverse it. If you can renature them if you do the opposite. If you take away the heavy metal, if you take away the strong acid, if you take away the heat. So we'll cover this. We'll continue this on Monday. Good luck. Those of you that went on the field trip, look for the quiz.